Today's seminar program will focus on cover crops in citrus, results from a 3-year project. The program includes an overview on practical considerations and impact on soil microbes by Dr. Sarah Strauss, improvements to nutrient availability, tree growth and root distribution by Dr. Davie Kadyampakeni, and increased weed suppression by Dr. Ramdas Kanissery, and cost benefits analysis by Shourish, Chakravarty, and Dr. Tara Wade. As you know, cover crops can improve water and nutrient retention, promote microbial activity, reduce weed growth and insect pests, and improve plant growth. As citrus greening can significantly impact root growth and nutrient and water uptake, the benefits from cover crops may be an additional strategy to improve citrus production and reduce fertilizer and water inputs. Dr. Strauss, it's all yours.
Thank you, Manji. Thank you guys for coming out today. So what we're gonna do this morning is we're gonna give you updates and sort of our general findings for this 3-year study that we've been doing now that just ended this harvest spring. This was funded by CRDF. And the basis of it was to really try and get some data on what happens to soils, what happens to nutrient availability, what happens to the trees when you're planting cover crops. And we also focus just on annual cover crop mixtures because surprisingly, at least to me, there wasn't a lot of scientific data that had been put together on some of those types of measurements when we're planting cover crops in citrus. And so we wanted to get some initial looks at what happens and start thinking about or trying to figure out what's happening so that we could maybe then make improvements. So I'm going to give you guys a bit of an overview of the study itself, a little bit of background about cover crops, and then as I am a soil microbiologist, I'm going to focus on the microbial components that we found with planting cover crops. After that, Dr. Davie Kadyampakeni, as Manji said, is going to talk more about some of the others, so soil nutrients and tree responses. Dr. Ramdas Kanissery is going to talk about the weed results that we found. And then wrapping things up, some economic analysis, cost benefit analysis by Dr. Shourish and Dr. Tara Wade will be at the end. And so all of us were working on this project together. So first off, I suspect that most of you guys know this, but just to make sure we're all on the same page, cover crops are crops planted to benefit the soil. They're not harvested for profit. They are not a new concept by any stretch of the imagination. This is a photo from a book that was taken in 1929. It's kind of hard to see, but that thing in the middle is a ruler with a guy standing next to it in a row middle of citrus grove with cover crops. So this is not a new concept. But it hasn't been used as much in recent years in citrus. It's becoming increasingly common practice for grains, cotton, corn, soybeans, some vegetable production. And that's really where the bulk of the research has occurred in the last, I would say, 15, 20 years, has really, on the research side of things, really focused on those crops. And so again, we haven't had that much information on what happens when we plant cover crops in perennial systems. In those vegetable and grain systems, cover crops are often planted during the fallow season, obviously something we don't have with citrus, and they're frequently combined with these conservation or no-till management practices. So why do people do this? We're going to go over some of these results that we found, but one is that they can reduce weeds. For when you've got grain fields and cotton, corn, soybean, you're planting during the fallow season, it can be really important for reducing soil erosion. In some soils, that's a very important method for reducing soil compaction. And then the big one, particularly for us here in South Florida, is that it can increase soil organic matter. And increasing soil organic matter is important because it can provide both nutrients, nitrogen in particular, and enhance that nutrient cycling through microbial activity, which I'll talk about in a minute, and can help increase soil moisture. And as you're all probably very, very aware, our soils here have very, very, very little soil organic matter. So this is I like this diagram that to try and illustrate all of the different benefits that can be found from adding organic matter. And so when we talk about adding organic matter, one of the main ways is through plant biomass. And so that's one of the reasons we were planting cover crops in the row middles rather than just having weeds, is it can provide a greater amount of actual plant biomass. And when you have that increased plant biomass, that's providing resources and material for soil microbes. And so that's going to increase your biological activity in your soil, possibly your diversity, which will then increase the amount of decomposition, which will release more nutrients from that plant material. As those nutrients are released, again, that's going to do all sorts of other things to your soils and microbial communities that could potentially improve your plant growth. And so again, building soil organic matter is primarily in our soils with cover crops happening through two ways. Again, this plant biomass, and then the cover crops themselves also are going to release exudates or different organic compounds from the roots that also are going to attract certain microbial organisms that will help with this entire cycling process. So it's a sort of a two-pronged approach. The plants themselves and the exudates that they might release, and the plant material itself, when it decomposes, is adding that material to the soil and enhancing some of that activity. The other big important component, or potentially important component when you're thinking about cover crops, as I mentioned before, is nitrogen. And so one of the primary ways that you can add some more nitrogen to your soil with cover crops is by planting legumes. And the way legumes are adding nitrogen to the soil is through the symbiotic relationship with a bacteria. These bacteria are called rhizobia. They colonize and attach to the legume root, and they are basically fixing nitrogen for the plant. It's a symbiotic relationship, and that these bacteria are going to fix nitrogen, and in return, the plant gives them carbon, gives them energy sources. And so, you may, if you've ever pulled up a cowpea or something like that, you see those little nodules on it. Those are the rhizobia nodules, and inside them are all of those bacteria. If you slice one open, there's a ton of bacteria in there. I think that this statistic I found that one nodule can contain up to 10 to the 9th rhizobia. So that's a huge amount in these nodules are not very big. And some bit of trivia that I always like to share about rhizobia is, if you notice in this picture of some that we sliced open, they look a little pink, and that's one way you can detect that, yes, there are active rhizobia in there. So nitrogen fixation is a process that requires no oxygen. And so one of the way that the bacteria in the plant are creating that low oxygen environment is to have this protein that buffers the oxygen, and it's called leg hemoglobin. And it's a very, very similar protein to what's in our blood. And so this is our sort of relationship between our kinship with rhizobia, which I always find interesting. The other trivia fact is that this is the same protein that's being used in the Beyond Beef products to make them look pink. So next time you're at trivia night, there you go. So, like I said, nitrogen is certainly a potential input when you're planting certain types of cover crops. But the other thing to keep in mind is that microbes are involved in all aspects of the nitrogen cycle. Every time you're applying nitrogen to your soil, microbes are doing something with it. And so it's important to remember that the microbes are very, very integral to the cycling of nitrogen. And so whether it's coming directly from those rhizobia that are attached to your legume, or whether it's from the nitrogen that's in those cover crops just that aren't even legumes, and that is being broken down by the microbes in the soil. The microbes are critical components. So there are a lot of different cover crops that you can plant. So I talked about legumes, and this is, I would say, still very much an ongoing discussion. Certainly, I think a lot of folks have probably maybe already tried or have some perennial peanuts as a cover crop in their groves. I know that's not too uncommon. A lot of folks do Bahia grass, which has also been shown to be a really nice cover crop, particularly with some pasture studies. One of the difficulties, though, particularly with the perennial peanut, is it can be hard to establish. It can take a little while to get established, and it's not cheap. And so for that reason, when we started this study and we're designing the study, because it was only 3 years and we wanted to be able to get some kind of data, we said, let's focus on annuals. But as you probably know, there's a huge variety of annual cover crops that you could plant. Cowpeas, sun hemp, vetches, clovers, all of those can provide some legume components or some nitrogen components. And then there's a wide variety of non-legumes that seem to do pretty well in our South Florida climates. And they can also provide slightly different benefits. So some of these might have more benefits and with nematodes. Some can provide different types of nutrient benefits. And so those are things to keep in mind when we're trying to design these mixes. Of course, like I said, again, this is still definitely an ongoing area, and trying to figure out what are the appropriate mixes and when to plant them is definitely something that we're working on. So the other discussion that often comes up in cover crop studies and research is whether it's good to plant a monoculture versus a polyculture. Monoculture being one single cover crop versus a mixture of cover crops. And there are really pros and cons to both of them, and it depends a little bit on what your main goals are. We've been focusing on doing polycultures on mixtures of cover crops, primarily because we are wanting to have staggered timings of germinations in an attempt to really have a better weed control coverage. And then to have mixtures of some of those cover crops. And some studies have been shown to have more benefits for that nutrient availability and nitrogen cycling, and that there's some competition between legumes and non-legumes that actually forces the legumes to fix more nitrogen. And so that's one of the reasons that we went with mixtures. But again, there can be benefits to having a monoculture as well. Things to keep in mind when you're thinking about planting annual cover crops. And so, you know, obviously the major place you're planting them is the row middles. In our study, we just did the row middles. We did not go trunk to trunk, but that is a possibility if you are interested. And then the other thing to think about is how to prepare your row middles before seeding. And so we've had a couple of different things where we have primarily for this trial to really try and ensure that we were getting a good cover crop germination and good stand. We were mowing and herbiciding the row middles before we planted. But we've also been doing some trials more recently where we have not been herbiciding and we're still getting good coverage. So it really kind of depends on how bad your weeds are when you start. And so that was the other thing that we've done too for some other trials where the first time we herbicided before planting, and then subsequently we have not been had to do that because we've had a really nice cover crop stand and had less of a weed presence. We've been using a no-chill seed drill to plant, trying not to disturb the citrus roots that are in those row middles. And then the other big key is that for annuals, you have to have multiple plantings per year. And so we are have been trying to time our plantings with the rainy season, which you all know is slightly tricky. But generally trying to get cover crops in the ground roughly right about now, sometime in June, which is obviously difficult to predict with the rains, and then trying to get a next planting in towards the end of the rainy season, end of October, beginning of November. This way, and unlike in vegetable or cotton, corn, soybean, where they can be very concerned with not wanting the cover crops to go to seed, because then that could be competition for their subsequent cash crop. That's less of a concern with us, and actually something that we kind of would like. We'd like to have those cover crops go to seed and bloom and produce seeds, because that could build up your seed bank so that maybe in future years you don't have to plant quite as many. And so that's why we've had this really long growing season for these cover crops, is to really try and build up that, allow those to go to seed and build up that seed bank. We have not done any studies yet to analyze how much of a seed bank we've built up, but that is the general idea. And so that cover crop that we plant in the end of October, November, carries through until harvest. Generally, at harvest, these were all on Valencia. So harvest activity does, trample some a bit, but we actually also usually have seen some stuff pop back up after harvest for another month or two before it gets really dry and we try and come back in and get the next planting in. All right, so what exactly did we have for our field trials? So we had 2 locations. Both are in the general Immokalee area. We're going to refer to them as the North and South Groves. They were managed by the same company, but they are in distinct locations. They were Valencia on Swingle, and these were pretty old trees. And so we had a number of different treatments. So we had 2 different mixes, a legume plus non-legume cover crop mix. And then we had the same non-legume cover crop mix, but without the legumes, because we really wanted to try and quantify what is this contribution of the legumes. We also had planned to do those same cover crop mixes with this treatment called eco-mowing. And this is something that's very common in Brazil. Excuse me, where when the row bundles are mowed, instead of the clippings just going right behind the mower, that they are sort of chucked underneath the trunks. The idea is then that even if you're not planting trunk to trunk, you're getting some of that biomass underneath, closer to the trees, and potentially helpful to the tree, and again, increasing that organic matter plant material, even without planting the cover crops. And so our idea was to try that. I'll talk to you about that a little bit in a minute. And then we had a grower standard control, which was just regular management, basically weeds that were mowed periodically. So what did we plant? We had a whole bunch of different things. And so, like I said, we had a summer planting, which was June-ish, and a winter planting, which was end of October, November. And so for legumes, generally sun hemp and cowpeas. In the winters, we did try some clovers. We never had great success with the clovers, to be honest. The non-legumes, we would use buckwheat, which was always nice because it's an early germinator. And so that would be one of the first things to come up and start hopefully providing some competition for weeds. Different millets, we did both Egyptian wheat some years or sorghum Sudan grass. And then the star sort of winter cover crop plantings very quickly became daikon radish, which you'll see some pictures of, is very, very impressive germination and consistently good germination regardless of the rains, which I thought was pretty impressive, as well as oats and rye. And so in these field trials, we made a whole slew of measurements. And so Dr. Kadyampakeni is going to talk to you about the tree growth and the fruit yield and the root growth and density measurements next. And like I said, Dr. Kanissery will talk about weed growth measurements, and Dr. Shourish and Dr. Wade will provide data on economic benefits. But I will share with you some results on soil organic matter and nitrogen and the soil microbial community. And so for these measurements, we were collecting samples once a year, basically in the middle of the summer, which is when there's a lot of root growth for citrus trees. And so that's where we wanted to try and capture some of that active microbial community that might be directly impacting the trees at that time. So we did find that soil organic matter, there were some increases with cover crops. And so in these graphs, I've got the North Grove and the South Grove, and you'll notice very quickly that there are some big differences between these 2 locations. And in blue are the data from before the trial started, so sort of our baseline, and in the red are the results after 3 years. And we've got the legume plus non-legume treatment, the non-legume, and the control. We had very little, that eco-mowing treatment that I mentioned, we actually had very few differences possibly because we also were not mowing that frequently. Very quickly after we started this trial, we realized, well, yeah, we don't want to mow our cover crops too frequently. We don't need to mow our cover crops very frequently. And so when we were doing that, it wasn't as often as I think we had planned. And then the few times that we did do it towards right before planting our next cover crops, we really just weren't seeing that many differences in the between treatments, particularly under the canopy or even in the row middles with that mowing style. And so I'm really just going to be presenting data here from the cover crop treatments because those are the ones where we saw some big impacts. So again, we've got our three different treatments here, and you notice that the baseline before the experiment even started in soil organic matter was very different between these groves. So South Grove had like barely 1%, and the North Grove already had about 2.5%, which is pretty high for our South Florida soils. So, but this is one of the reasons why we wanted two locations, is to really, because your starting conditions can certainly impact how these treatments will perform. But despite that, despite that higher start in the North Grove, we still did see a bit of an increase in soil organic matter, particularly with the cover crop treatments, both the legume, non-legume, and the non-legume only compared to the control. It was only about 0.5%. But we did see a slight increase. There was no statistical difference in the soil organic matter between these treatments in the South Grove. And some of that is we also had a lot of difficulty with some of the germination at the South Grove trial. The canopies weren't as big, and so I think there was a lot more sun getting into some of those row middles. Again, maybe could be due to the lower soil organic matter starting conditions. Lots of things that we're still trying to assess and figure out on why those differences were occurring, but obviously some big differences between the two locations. We also did find some changes in soil nitrogen with the cover crop mixes, particularly with the legumes. Measuring these are data on nitrate and ammonia, which, as you might know, can be a little tricky to measure in soils. The concentrations of these two are highly dependent on the moisture and how much rain you've recently had. And so again, we were collecting these measurements in the summer. So it's a little problematic on that. And you can sometimes get a lot of variability within a grove, as you can see with some of these big error bars, and between years that may not be related to a treatment. And so we have to sort of keep that in mind when we're looking at this data. But we are still seeing increases, particularly in ammonium with these cover crop treatments. Technically, the legume, non-legume has more ammonium than the other treatments, particularly in the North Grove. Again, not big differences in the South Grove, potentially related to that germination. Also see changes in nitrate, but again, this is a little trickier to measure in the soils because, as you probably know, nitrate is very mobile. And in the summer, we've got lots of rain and water and leaching. And so looking at those numbers is not an absolute, I would say. That said, what I get particularly excited about, because I'm a microbiologist, is we saw some really interesting things with the soil microbial community. And so these graphs I've actually broken down by year because I find this really, really impressive. And so this is looking at the total bacteria and archaea abundances in soils. And so here again, we have the legume, non-legume, the non-legume cover crops, and the grower standard control. And so the blue here is before the trial started, and then the 3 years. And you can see that in the grower standard control, there was really no change in those total bacterial numbers. But in these legume plus non-legume treatments, we saw massive increases in the number of bacteria and archaea, and that continued to grow throughout the 3 years. We're also seeing changes in the composition of these bacterial communities. And so if you've never seen one of my talks before, these plots are, each dot represents the total bacterial community composition of one sample. And so the closer together dots, triangles, whatever shape are to each other, the more similar that composition is. And so, for example, here we've got all of these squares, regardless of color, all clustered together. And so this means that in the North Grove at the beginning of the trial, there was a similar bacterial community composition for all the soils and all the treatments. That's what you want, right? And then by the time the trial ended, you can see that all of the control soils have a very different bacterial community composition than those with the cover crops. We also saw very similar community composition at the beginning for the South Grove, but not as big of a difference, well, again, between the cover crop treatments, which are in red and green, and the control that are in blue in the South Grove. Not only do we see changes in the composition, but we're seeing changes in the activity of some of these organisms. And so remember I said that the microbes are very important in the nitrogen cycle. And so if we look at mineralization, which is the transformation of organic nitrogen in plant material or in soil organic matter in other microbes into ammonium. So this is a process that is performed by a huge number of bacteria and organisms. If we measure that, we actually found that there was a much greater rate of mineralization in the soils that had the legume and non-legume cover crops. We still saw more in the non-legumes again. So there are more of these organisms that are able to take nitrogen from other places, other materials, and transform it into ammonia that could then be used by other organisms or the tree. And so there's this change in the activity of these organisms under these treatments. We also see changes in nitrification, which is the process of ammonium to nitrate. And this is performed by both bacteria and archaea. And so if you look down here, this is the number of, or the abundance of the bacteria that perform this process and the abundance of the archaea that perform this process. You can see that in the legume plus non-legume treatment, we have more of these organisms, and we also have a greater activity of these organisms. So if we look at the rate of nitrification, the rate of activity of these organisms, we see there's much more nitrification activity occurring in soils that have these legume and non-legume cover crops. So again, more activity, more of this nitrogen is being cycled through the soils that have these types of cover crops. We also saw really, again, to my mind, I think fascinating changes in denitrification. So denitrification, there are a bunch of different steps to it. So one of the first steps goes from NO2 to NO. And we see very different organisms. This is done by organisms that have this particular gene. And you can see that we have completely different organisms under the control soils compared to the cover crop soils that are doing that part of denitrification. Similarly, for the other part of denitrification going from N2O to N2, we have very, very completely different organisms that are doing that based on the types of cover crops. So not only just cover crops in general, but you've got different denitrification occurring under legume plus non-legume versus non-legume cover crops. And we are also able to measure differences in the amount of gases that are produced. And so again, there's less N2O being emitted from the soils that have these legume plus non-legume cover crops compared to the other treatments. So again, this complete change in the bacterial community composition and the activity of these organisms. We also are seeing changes in the fungal community composition. And so this is brand new data that we have just started analyzing. But I think it's really interesting here that, again, this is from the North Grove. At the very beginning of the trial, the fungal community composition was pretty similar. And you can see the circles in here are also in the same circle with the squares. This is indicating that by the end of the trial, the fungal community and the controls really hadn't changed. So after 3 years, that fungal community and the control soils was pretty stable. But underneath cover crops, we've got big differences in the fungal community composition, again, based on what types of cover crop mixes are planted. Again, not as many differences between the fungal community and the treatments in the South Grove. This is something, like I said, we've just started looking at, and I'm really excited to dive into a little bit more and see what might be happening with these fungal communities. So just to quick summarize, I think one of the big lessons that we learned the hard way is the timing of planting of your cover crops is really important. Trying to really get those seeds in the ground right when it's about to rain is critical. planting mixtures of these annual cover crops did have some changes and impacts on soil organic matter. This is an area that I have a new study that's looking at a little bit more closely, whether looking at soil organic matter is the best way to measure in carbon storage in these soils. There might be some other measurements that are more appropriate for our subtropical hot, humid climates, and that, are we looking at the best ways to really try and gauge how much carbon might be entering our soils, how much organic matter. But it also had a really big impact on the microbial abundance. And I think the other is that we are seeing evidence, we saw some pretty strong evidence that the cover crops and the types of cover crops that you're planting have a really big impact on that soil microbial composition and nitrogen cycling activity. I'm gonna not spoil things too much for Dr. Kadyampakeni, but I think this is important to keep in mind that we are seeing some changes in these soils and these soil activities. How quickly those changes will translate to the trees is something that is less clear. But we are seeing impacts of these cover crops on that soil, which I think is a really promising, at least first step. So I'm really quickly going to thank a couple of people, obviously my collaborators on this project, but I have to really thank former postdoc in my lab, Dr. Antonio Castellano-Hinoso, who is now back in Spain, but he did a lot of fantastic work on this project. And I also would like to thank the CRDF for funding us. Think we might have time for a quick question. If not, I will be here all morning. Thank you. Yes, sir.
It seems like as the last 2 years have progressed,
yes, I believe Dr. Kadyampakeni is going to talk about that. Oh, I'm sorry. Yes, the question was, did we look at soil moisture impacts? And I believe Dr. Kadyampakeni will be discussing that. But that is a good question.
Then I have another question.
Good question. The question is if we looked at crimping or roller crimping for crop termination. We did not. Like I said, we really were just letting these grow to seed, and then we mowed them and planted the next one right into that. So, but we haven't talked about it.
Question from online. Sure. You planted everything no-till?
Pardon?
You planted everything no-till?
Correct. Yes, we planted everything no-till.
And did you have a local seeder?
The grove that we were working with had their own no-till drill, and then we have one at the center that we're using for some other trials. Yes.
Okay.
Any other questions? Great. Thank you so much.
So the next presentation will be by Dr. Davie Kadyampakeni. He is a soil scientist located at the Strauss Research and Education Center in Lake Alfred. Go ahead.
Thank you so much, Monji. Can you hear me?
Yes, we can.
Oh, thank you. So thank you so much to Dr. Sarah Strauss. She went over the overview of the project, so I'll not repeat myself completely, but I'll try to offer highlights. So my apologies, I have a lot of heavy data to present because most of it were the measurements that we did. But one thing I want to mention, there was a question on the water storage. So unfortunately, I didn't include a slide on water storage. We did see any differences, largely because of the sites we were dealing with, very high water table at the two groves. So we did see any differences in water storage. So that is something that maybe we might have to consider in future research, especially where our well-drained sandy soils like on the ridge. That might be a key factor to look at. But on the southwest Florida sites that we started, we didn't see any differences in water storage. That's something I wanted to catch, but we did not. We used sensors, but we didn't capture that in all the treatments between leguminous cover crops and also non-leguminous cover crops and the control. So I'll be talking about improvements to nutrient availability, tree growth, and root distribution. And I'm located in Lake Alfred, working as a soil and water scientist, as Monji rightly mentioned. So in this presentation, I'll cover a little bit about the tree growth measurements we did, the leaf nutrient content we measured, soil nutrient contents, and look at the harvest and food quality variables. I'll also look at the root health parameters, and then of course provide some conclusions. And I think there's room to speculate what we need to do going forward from the work we have done, because this was 3-year data, but most of the variables we evaluated didn't come out significant. But I think there's some indication of what we could do going forward to further elucidate some of these research observations that we have captured. So Dr. Strauss tackled a little bit about the benefits, and I want to make sure that that message sinks to all of us, that we know that cover crops, from the people who have started it, from the researchers in the Midwest, across the world, we have the good story of improved nutrient cycling, and we have heard those claims of better pest management, whereby you are controlling the agroecology, you're minimizing the pressure of pests, and once you change that, then you have less impact. So we thought this would be something to evaluate here, that would limit maybe still pressure and other pests. And then I know Dr. Kanissery will tackle this, but I think cover crops are also well known to suppress and control weeds. So that's something that we thought we could also look at. And then sustaining water quality, whereby you are having the cover crops improving the biomass and organic matter accumulation, thereby improving water retention of water and also nutrients. And obviously, something that we have also learned in other cropping systems is that cover crops help improve yield and growth. So this is something also we try to tackle in this project to see if we can get that also in citrus production systems. But actually, most of this work has been done in seasonal crops. So in corn, in peanut production systems and soybean, but not much in citrus. So this was a big opportunity that I think CRDF granted us to try to delve into this kind of research and come up with some better understanding of what we need to do with cover cropping. So on the right, you can see one of the raw meadows with cover crops, and then these are some of the tools we're using for measurements. So I'll quickly go into what we were looking at. So the first variable we're looking at was actually tree growth. So we were looking at tree growth variables, looking at trunk diameter, looking at the tree height, and looking at the stem circumference. And then we are also looking at the canopy volume and area. This was done every 6 months, very heavy data collection. And we thought that would help us, because usually we could do it once a year, but what we have learned is that when you do in spring, for example, before harvest, you tend to have a little bit of a heavier canopy. And when you take those measurements somewhere in fall, then you tend to have a little bit of a lesser, denser canopy. So we wanted to see those effects happening over time. So when it came to looking at the canopy volume, so our canopy volume measured in cubic meters, we didn't see any much changes in canopy between treatments in the North Grove. So as you can see here, we are having the dark line as the control, no cover crop, and then the blue line is the legume, and then the orange line is the non-legume cover crops. So we just split those into 3 categories. And what we are seeing there is that in the North Grove on the top, we didn't see much difference over time. So from October 2018, when we started the measurements, all the way to August 2021. In the South Grove, just one time point in March 2019, where we saw some difference, whereby we found that the control and where we had the legume showed superior canopy volume compared to the non-legume. But throughout the other times, we didn't see any difference. So very interesting results, just to observe that these were mature trees, as Dr. Strauss mentioned. They were planted in 1991. So maybe that also, that age, we don't see much difference because of the age. Maybe if it was younger trees, that we could capture that. When you look at the stem circumference, so kind of similar trend, no much changes in the south grove, but in the north grove, the control stayed like on top of the stem circumference. So looking at the time point of March 2019 and August 2020, February 2021, and August 2021. Yeah, so the dark line is the one for the control, the blue one is for the legume, and then the orange lines are the non-legumes, and the error bars denote the what we call standard errors or standard deviation. When you look at the canopy area, so that is more or less like the spread of the canopy, looking at the east, west, and north-south directions, we also didn't see much difference, but in the North Grove, we found some kind of temporal changes in canopy area. So in March 2019, in August 2020, in February 2021, and also in August 2021. So maybe towards the end, we're seeing more changes in terms of canopy area, but that was mostly in the control. And then there were still most of the top points that we didn't see much of the difference between the leguminous and non-legume cover crops compared to the control. So we try to now just look at the overall changes over time. So look at all those treatments. So you see the dark lines are the no cover crop, the blue lines are the non-legume, and the orange lines are the legumes. So as you can see, for the North Grove in the top, the non-legumes, the cereal cover crops, and the control are kind of similar. And then, of course, the non-legume and the legume were also having similar kind of areas. At the bottom, the legume was a little bit superior for the South Grove, was a bit superior in canopy area compared to the non-legume and no cover crop. So kind of confusing to conclude, because the one side you see something else, and the other side you see something a little bit different. And then when we looked at the canopy volume, we found that in the North Grove, so kind of most of the control is the dark line, the blue one is the non-legume, and the orange one is the legume. No significant changes, but we noticed that for the North Grove, the control was a little bit superior, but that went with mostly the background sizes of the trees. And the South Grove, or the legume and non-legume, a little bit superior compared to the control. With regard to stem circumference, kind of similar trend to the canopy area. So that was to do with tree growth variables. And then we went ahead and tried to look at nutrient accumulation in the leaves, because that gives us an indication of what's going on in terms of adequacy of how the tree is utilizing nutrients. So we did collect leaf tissue nutrients once a year in August. And then we did that following what we call University of Florida IFAS recommendations, taking leaves that are between 4 to 6 months old. And we were taking samples in 4 quadrants. So the northwest, northeast, and southeast and southwest portions of the tree to make sure we come up with a composite sample that's representative. And we use standard methods. We call that leaf dry ash digestion. It's quite common. That's what they do in the commercial labs and also in our IFAS labs. And we also looked at the carbon-nitrogen ratio for determining, carbon nitrogen ratio for determining the nitrogen content of the nitrogen content of the leaves by what we call a combustion method. So this is a little bit of a busier slide, but bear with me because I just want to capture everything side by side so you can have an idea of what we're talking about. Yeah, so the first panel is on the left is the one for nitrogen. The middle panel is the one for phosphorus, and the right panel is the one for calcium. And the top panel is for the one for the North Grove, and the bottom panel is for the South Grove. So not much difference in the leaf nutrient composition for nitrogen and phosphorus, and even for calcium. So in the North Grove, but in the South, we saw some difference in the nitrogen and also in the phosphorus. For some reason, as you can see here, well, we noticed that for the South Grove, the nitrogen was a little bit lower in the non-legume, but it was the legume and the no-cover crop were quite similar. In the South Grove, also for the phosphorus, as you can see, the non-legume had a little bit of elevated phosphorus, but very subtle differences. As you can see, the p-value are using 0.1%. So that's a little bit of not a conservative significance, but we're still trying to delve into understanding if there are any little differences. No much difference in calcium, and this tends to drive me a little bit into kind of confusion, because I was anticipating to see some high levels of calcium or in some treatments. But what we have noticed when we did the soil tests, we tend to have high and elevated levels of calcium in those soils in both the North and South Grove. So maybe that explains it, that we didn't see any differences. And then we also looked at some of the micronutrients. So in this case, I'm just showing something on boron, manganese, and zinc. So no differences in both sides, no differences between legumes, no cover crop and non-legumes. So kind of similar trend to some of the treatments observed in the macronutrients. So for the top level, it's the top panel is the one for the North Grove. The bottom panel is the one for the South Grove. So as you can see, the dark line is for the no cover crop. The blue line is for the legumes, and then the orange line, the orange bars are for the non-legumes. And then another important aspect that we looked at was to look at the nutrient analysis. So we sampled there once a year in August, and then we had 10 randomly selected trees in each plot. And we sampled those trees and took a sample and composited that. And then we had the one location in the raw meadow to sample from where we had the cover crops. And then, and then we had the side that was under the canopy, which was around 1 foot from the tree trunk where we sampled. So we wanted to stay within the wetted zone. And then all those soil samples were taken in the top 1 foot. So we got that sample right there so to make sure that we are in the active root zone. And we had about a total of 2 samples per rep. So under the canopy and within the raw meadow. And then what we did with those soil samples, we analyzed for inorganic nitrogen. So I think Dr. Strauss also mentioned about ammonium and nitrate. Those are the inorganic nitrogen forms that we looked at as well, using the what they call 2-molar potassium chloride analysis. And then we also looked at soil macro and micronutrients, focusing on phosphorus, calcium, potassium, and then we also looked at the micronutrients, boron, zinc, and manganese. So right there, we have the panels for the ammonium on the left, nitrate on the middle, and then we have phosphorus in the far right. And at the top level, we see the North Grove, and then the bottom panel is the South Grove. The grayish panel graph is the one for the control. The blue graph is the one for the legume, and then the orange one is for the non-legume. Yeah, so as you can see, not much difference. I think that's one thing also Dr. Strauss mentioned. Not much change in ammonium, nitrate, and phosphorus, and the canopy and between the raw meadows. So this was part of when we tried to look at the data composited. So that quite very surprising. We wanted to see if we could capture differences in those treatments, but mostly no differences between those treatments. And when we looked at the other macronutrients, so looking at medically available phosphorus, magnesium, potassium, and calcium, because some of these drive, root health, drive tree canopy, drive yield, but we found that mostly the results are comparable. But one thing that I want you to work with me is, I kind of messed up this a little bit. Yeah, I would like you to work with me here a little bit. So the top panel is for calcium, the next panel is for potassium, the next panel, second from the bottom is for magnesium, and then the very bottom panel is for phosphorus. And we have this data analyzed by year, so 2018, 2019, 2020, and 2021. Four solid years, or 4 sampling times for the 3 years of the project. This was the starting point. So the panel at the left, so the left graphs are for the north graph, the right graphs are for the south graph. So one thing I would like you to notice is that, as I mentioned earlier, was that I'm using a very conservative, not a conservative p-value, so 0.1, but I wanted to still delve into this. So for the north graph, because we tended to have high organic matter, as Dr. Strauss also shared there, somehow we had better numbers for the calcium. As you can see, the numbers I was showing, the leaf values, that we didn't have much significance in terms of calcium content in the leaves. You can see the values way close to either 2,000 and close to 4,000 parts per million of calcium. The phosphorus, the potassium was reasonable. Those are the ranges at both sides were a little bit reasonable to what we expect. And then the magnesium was also a little bit on the higher side, on the north graph, so between 200 to 400 parts per million. And then the phosphorus was even on the north graph a little bit beyond the excessive levels that we normally see in our recommendations, so over 500. So most of these nutrients are residual nutrients. That's what I would speculate. And on the north graph, we tended to see those high numbers. And the south graph, a little bit different, lower numbers compared to the north graph. And then, as you can see where we had the stars, that's where we're able to capture some differences. So under the canopy, we tended to see a little bit more elevated nutrients than in the raw meadows in this case, except I think on the south graph where we had in the year 2020, where we saw that the raw meadows and under canopy were quite comparable. So in terms of yield, I would like to share this a little bit. We did this once a year. We collected 10 fruits per plot just to use that for juice quality analysis. And then we looked at fruit size, we looked at the diameter of the fruit, or vertical or horizontal. We looked at the fruit quality, but I will not share all this that I'm just trying to highlight what we mentioned, but I'll share some of the juice quality variables and also fruit weight variables. Yeah, so there we go with the juice content. So the acidity was quite similar in 2020. We couldn't measure the fruit yield in the south graph because of COVID restrictions, so we couldn't go there. We had some limitations from IFAS, so couldn't go to that side. But as you can see, not much difference. And even this year, we are analyzing the data for 2022, but not much difference between treatments in terms of acidity. And then also, when you looked at the juice in terms of pounds per box or total soluble solids. So you can see on the left is the juice in terms of pounds per box. On the right is total soluble solids. Not much difference between treatments, as you can see, between Nova, cover crop, legumes, or non-legumes. Quite similar performance across all the variables. And when we looked at the harvest, we didn't see difference in fruit weight. So we did these yields. I was able to get the yield values for this year to include in here. So as you can see, so on the left, that's where you can see the harvest in terms of pounds per tree. And then at the bottom, you can see the no cover crop, legume, or non-legume. Yeah, so not much difference in terms of yield. In 2022, we're not able to get the no cover crop. I think the pickers missed, they picked from our treatments. They thought that was not part of the study. So that's why we have that missing point there. Yeah, but very interesting to see that we were not able to capture differences in yield per tree and even I think per block in boxes per acre over the duration of this study. Another last variable that I want to share with you is the aspect of root growth. So I just have this schematic that I love so much that was developed by Dr. Ali Atta. He's a postdoc right there in Immokalee, and this was a schematic for one of the papers we co-authored. We had installed these tubes at around 20 inches from the tree, and then within the about 20 inches from the emitter. And then it was within the zone at 45 degrees. And we inserted that tube in the under the tree canopy. And so this is where you have the main line, and then we have the microjet there. And then I would like to just share with you some of the highlights of what we observed. So I had this evaluated for root volume, root length, root area, and we also had some root dieback estimates and root longevity, but for the sake of time, I just put a few slides. So what I'm showing here is for the south grove only. We saw that we had better. So for example, here for this, excuse me, I kind of missed this one. Yeah, so here I presented data by 4 treatments. So C1 is where we just had the legume, and then C1 was where we had the legume mixed with a non-legume. C2 was a non-legume, and then C2 was a non-legume mixed with a legume, and then we had a standard control. Yeah, so largely we found that where we had the leguminous cover crops, either in a mix or just a leguminous cover crop, we found better growth. So this is the blue one and the orange one, better growth of the roots. And something also for the length. So that was something that gave us some encouragement that we might be seeing better response over in terms of root growth compared to having cereal crops. So this was something that we noticed, and we take those measurements every quarter. So for March 2019, when we installed the tubes, all the way to February 2021, when we did the last measurements. I think we did those measurements in February 2021, and we did another set of measurements, but I've not been able to process the other data sets that we did last. I think we completed the analysis last time in October 2021. So hopefully in the next few months, when you do publications, we'll have some of those data shared. So what have we learned so far? We noticed that there were no changes in fruit yield, juice quality, kind of area, kind of volume and stem circumference and leaf nutrient content over the 3 years. However, as we have learned in even other studies we've been doing in the state, changes in production due to cover crops in the perennial cropping seasons usually take a little bit of time. And I'm suspecting in this case, might be more than 3 years. And one thing we also learned was that root growth trends showed greater root length and volume in legume cover cropping systems over cereal-based cover cropping systems and the control. So at this point, I would like also to share with you that maybe there's something to consider, that maybe we need to understand the root growth and water dynamics better, because we didn't see any differences in the water dynamics. That's why I didn't even want to share, because they're just actually similar trends because of a high water table at both sides. We had a very elevated water table within about 2 feet from the topsoil. So that affected our moisture content. And then also, I think there's no room to understand, because these were mature trees, close to 30 years old. We can also maybe do further research on young and bearing trees. And then of course, other mature trees, but that are younger, maybe 10 years, 15 years, to provide necessary information for adjusting cropping practices for our commercial growers. And then also, we might, Dr. Strauss mentioned about the various cover crop species, which is very important. We need to have that evaluated in the flatwoods, in the southeast, and of course also the southwest, as we did, and the ridge regions. And that should be done over time. So to identify agroecological trends, the right mixes, the right species that fit our growing regions for citrus in the state. So at this point, I'd like to join everyone in thanking the University of Florida and our department and the experiment station for allowing us to do this work, and also the Citrus Research and Development Foundation for funding this research, and my project team, and obviously, of course, my collaborators in Immokalee that we Dr. Strauss and others that helped us do this work. At this point, I'll be happy to take any question, and my contact phone number and email are here. Thank you.
Any question?
Yeah, hey Davie, how old were the trees and were the trees hedged during the trial?
Oh, thank you so much. The trees were planted in 1991, as Dr. Strauss mentioned. That's why I didn't repeat in my methods. So we agree that don't repeat whatever is already be highlighted. They were about 31 years old. When we started this trial, they were about close to 27, 28 years old. Now, but they were planted in 1991. So now they're about 31 years old. Oh, they're not hedged as we did the trial. No. Thank you.
Davie, it looks like all the energy went to the roots.
Yeah, that's very interesting. And I was not happy not to see the effect of the root growth on the yield. I wanted to see the yield change, but I think sometimes some of these treatments are a little bit stubborn, as Dr. Strauss mentioned. And I've seen that happen even in some of our studies where we have been changing the macronutrients. We have done some studies for about 3 years and not seen a response until maybe the 4th year. That's when I've been able to see a response. So I'm suspecting that that might be the case, because Dr. Strauss showed the accumulation of the organic matter in some of those treatments, but we haven't seen that tree go into the tree yet. So it might take a bit of time for mature trees to show that response. Yeah, so it seems the roots benefited. Yeah, but the moisture didn't change much, which was interesting. But I think maybe if we do that on a well-drained sandy soil, we might see something different, but we don't have those data yet.
Hey, Davie, the legume cover crop, was that once a year or was it multiple seedings? And was it terminated or left to go to seed?
Yeah, so that's a good question. That one, Dr. Strauss mentioned, the way we were planting twice a year. So in summer and in fall, like summer and winter. So that's how we're doing it. So the legume cover crops or cereal, none of those were perennial. So we're planting those in summer once, and then plant again in fall once. That's what we're doing. And then we let the seed sit there, and the farmers sit there.
Do you crimp it at the end or mow it or disc it or between those two cover crops? You mow it.
I missed that question.
He was asking about how the cover crops are terminated.
Oh, I see. Okay, thank you.
Thank you, Davie.
All right, thank you so much, everyone, for your time.
Okay. Next presentation is by Dr. Ramdas Kanissery. He's a weed scientist working at the Immokalee IFAS Center.
Hello. Hi. Morning. So I'm Ramdas Kanissery. I'm a weed scientist here at the center. So I was also collaborating with Dr. Strauss, Dr. Davie, and Wade in this project. But one of my major role is to look more closely into how cover crops can contribute to weed suppression in citrus growth. I'll give you a brief overview and set the context for this talk. So I was showing these two pictures, typical weed control scenarios from a citrus growth. On the right, on the left, we have a perfectly clean or well weed managed drip line or the under tree or tree row area, which is ideal because we can reduce the competition as much as possible. And on the right, we have some situations where, this often occurs from time to time, we not be able to keep it clean. So weeds can encroach into your drip line and the under tree area, especially in Southwest because of the weather conditions, rainfall and things like that. But I want to talk to you about this thing. This message I want to give you all, not only because I'm a weed scientist, I really think managing the weeds in the tree rows really matters because that's what we see from our studies. For example, a graph like this, I want to show, if you can control your weeds under the tree rows, and 75% of the weeds are controlled, we can get up to 25% of increase in the yield. And again, this is all about competition. Whatever you are doing to boost your tree has to reach the tree, not be stealing away by the weeds, right? So competition is one thing. Another thing, keeping it clean, reducing the weed pressure also help improve your pest and disease management efforts as well. Because one of our studies, we show that some of the weeds like Spanish beetle, dog fennel, or primrose can actually support psyllid growth in citrus fruits. So keeping your weeds really has a lot of benefits. So that's why we typically spend a good part of our budget, 12 to 15% of the production goes towards weed control. And these days, herbicides prices are increasing. There is inflation. Then also there's a supply chain issue, so lack of availability of herbicides. A lot of things are in the play, but still, growers try to manage their weeds effectively, and that they can see a benefit in the production. I want to invite your attention to this picture. So when I talk about weed control, as a weed scientist, I will add a lot of things in the weed control toolbox or strategies. So one thing I always tell is sanitation or prevention. So first thing that we need to do in a weed control in citrus is preventing the weeds to come into your tree rows. And it all starts from here. So our research shows that more than 50% of the source of weeds in tree rows is from the row middle. As you can see, if you don't keep our row middle vegetation tidy, if you don't mow it in time before setting seeds, flowers and seeds, highly likely it's going to spread into your tree rows and making your under tree weed control, which is more important, very significant, very difficult. So I know with this context, I would like to add alongside several other strategies in the weed control toolbox in citrus, like prevention or mechanical or chemical, which is the most popular. I'd like to add cover cropping to this toolbox. It's an excellent tool, a very effective tool for suppressing weeds in your grow. And I like to tag it as a cultural practice of weed control. So as Dr. Strauss clearly pointed out, cover cropping is not a new practice. It's been everywhere, especially for weed control. For example, it was being practiced in several production systems, several scales, and things like that. I was a weed scientist I had spent some years in Illinois in the corn and soybean jungle, and we used cover crops there. For example, on the left side, you can see cover crops planted in a standing corn crop in the row middle to suppress weeds. Sometimes cover crops are used with the standing crop, or sometimes it's used in the fallow season when crops are not used. But cover crop used been everywhere. I mean, several production systems. Also on the left, this is a word from on the right actually. This is a word from our IFAS, where we are trying to promote some cover crops in raised or beds, vegetable beds, small scale, sometimes kitchen garden, to suppress the weed. So, anyways, my point here is it's been everywhere. It's been in use in several scales and several production systems for weed management. But for citrus, it's kind of new, and it's an emerging practice, as Sarah pointed out. There are several benefits, okay? But we already talked about that. I don't want to go any detail into that. But my focus here is suppressing weed growth. And I really think it's a great tool in the toolbox that is provided by our Mother Nature, weed suppression. So let me talk to you a little bit about how cover crops manages weeds, how it suppresses weeds, or mechanisms involved in the weed suppression by cover crops. So first, there are several mechanisms, but I'm going to talk about a couple that is kind of relevant to our observations. Shade. I think shade is a natural weed killer. As you can see, when we select those cover crops which can grow faster and have a good canopy and close up that canopy closure keeps the light out from reaching the soil surface and keeping the soil cool. Not only that, it will prevent the weeds to emerge. For example, this is a picture which was taken under the radish, daikon radish, which was a major cover crop that germinated and predominantly found in our mix. And you can see under the canopy of the radish, completely shade. And as you all know, most weeds do not like shade. They will not germinate there. There may be seeds in the soil, but they don't want to germinate because of the shade. That's one mechanism. Okay. The second mechanism is plants talk to each other. Likewise, cover crop can talk to their neighbor through allelopathy. They can tell the neighbor, don't grow here. So we found many of our cover crops that we use in our mix. Again, has this a particular phenomenon known as allelopathy, which means it can release a type of biochemical into the surrounding through the soil or through the air, and it can suppress the germination and growth of the weeds in the nearby area. So I want to point out here, this process not only occurs when the cover crop is living, but also this occurs when they are terminated and incorporated back into the soil. So timely termination is very important here. We have to make sure all the benefits of allelopathy is obtained, for weed suppression when we terminate that back into the soil. So that is another mechanism. So these two mechanisms I want to keep you in the back of your mind when we talk about weed suppression. Okay. So now let me show some of the observations we found in our study. So this is a general, an overall picture that I want to show. Let me set this graph for you. So on the top, we have growing seasons or year. And we also have two study locations, okay, 1 and 2, top growth and south growth. So on the x-axis, we have the treatments like non-cover crop, legume, non-legume. And on the y-axis, we have the weed density, number of weeds per square meter. So in our data collection, in most of the seasons, in most of the locations, except a couple of exceptions, if you look at the no cover crop, the cover crop, whether it's legume mix or non-legume mix, it considerably, significantly reduce the weed. If you look at it closer, I want to pick two seasons, one location and 2 years here. The mix 1 and mix 2. Mix 1 is with the legume and mix 2 is without legume. Then we have the same thing with eco-moving and non-eco-moving. That we did not talk a lot here, but it was you're trying to put the middle vegetation when we move it under the tree canopy to see if it's spreading any weeds into the canopy. But it looks like it was very similar to what we tested in the raw materials also in terms of weed suppression. But my point here is we found when you compare the global standard or non-cover control, there was 80% reduction in the weed density in the row. This is pretty clear from this type of data. When we look at the type of the weeds that's been suppressed, which is most of the troublesome weeds in our area, that includes guinea grass, very problematic weed, crabgrass, and sedges. Right? And one thing I want to point out here, guinea grass is a grass that we see a lot in the raw material. Ultimately, when we try to mold the raw material vegetation, when we try to maintain it, ultimately it might end up as a soil culture of guinea, right? And this is a very problematic weed because it's going to get a lot of seeds, and these seeds are going to deposit in the 100-feet area as well. So some of the grasses and sedges, again, we have annual sedges and perennial sedges, including yellow nut sedge, purple nut sedge, all those were included in the suppressed weed. Not only grasses or sedges, we have several broadleaf weeds such as Virginia pepperweed, ragweed, parthenia. This weed is considered as one of the most troublesome weed in South Florida. It will not respond very well to paraquat or remoxy. It needs some specific selective herbicide. And nightshade, lake nightshade, another species of broadleaf is very popular in products of the South Florida. So these are all the weeds in that spectrum of weeds controlled in the raw middle by cover crop in our study. So some of the lessons learned in our study, okay, this 3-year evaluation. The weed suppression is really, correlated to the amount or density or biomass of the cover crop. It's all about biomass. It's all about density. For example, if you take a look at this piece of data where you have the cover crops per square meter, density of cover crops on the y-axis and the x-axis weeds, number of weeds, you can see a strong negative correlation. The more the cover crop, the more weeds are. Okay, this is like a pooled data from different locations in different seasons. I want to show you that correlation, but the point here again is shade, right? More density, more lush growth of cover crop, more shade, and more weed suppression. So the emergence and maintenance and a good cover crop stand is very important to get a good weed suppression. Now, talking about the weeds, the cover crop germination, cover crop density, an interesting trend I want to show you here. Again, we have different seasons as well as years as well as locations, like the last graph. And on the y-axis, we have the cover crop density, number of cover crops per square meter, and the cover crop treatment, different treatments in legume mix and non-legume mix. In one location, there was an interesting trend. All the 3 years, consistently, the legume mix, which had, I believe, sun hemp, clover, and cowpea, God's mix was germinating better, or the stand was better, a little bit more better than the other one, but the other location that one was not. Anyways, I want to show that in among all the cover crops that germinated and grown in our raw mill, more than 60 to 80%, depending on the location and season, daikon radish was predominant. And this is a picture of a small pond from our experiment. So on the left, you have a cover crop area and lush growth of daikon radish. And on the right, we have a grower standard which was maintained like a grower standard, the control in the raw middle. And you can see all sort of nasty weeds that are coming up like guinea grass, black nightshade, geranium. Okay. So the daikon radish, I want to talk a couple of more points about daikon radish because since it was our predominant species in our mix, these are just the tip of iceberg. They have a lot of underground growth, and we found that in 60 days it can grow up to 3 feet down deep. This is very important, especially Sarah to mention the soil, holding up the soil, which is very important in our sandy soil here, and also improve soil aggregation and things like that. So apart from the weed suppression, daikon radish is going to give a little bit of soil quality improvement benefits as well. One other thing I really want to focus more in the near future is there are a lot of talks about biochemistry. So if you're not familiar with biochemistry, we have chemical fumigants we use in vegetable production and the production system, nematodes, weeds, and diseases. But we can produce fumigant naturally. And for example, mustard is a plant that's been used a lot for that type of studies. And guess what? Radish, daikon radish, close relative of mustard, and it's the same species or genus of Brassica. So this material or this plant from its biomass is known to release a compound known as glucosinolates, which is a sulfur-containing material, and it can be performed or act as a biostimulant. For example, there are several studies that have been occurring everywhere where they chop up the daikon radish and if that's something that's been used and spread inside the greenhouse or an enclosed area to produce that fumigation and things like that. So apart from the weed suppression activity, having cover crops like daikon radish may have some insect suppression as well as nematode suppression or fungi suppression activities as well. But again, this is something that we need to look further. Again, I want to show you how much daikon radish we have in that lush growth in the raw middle. Then one point also I want to bring here is the composition of raw materials. For example, we looked at what are the vegetations we have in raw materials in a cover crop area and non-cover crop area. So on the left, we have with cover crops, it's mostly cover crop, and we have a little bit of panicum, Virginia pepper, parthenia, but not cover crop compound. It was completely or entirely taken over by sedges and grasses. See, so if you ask me, when it comes to competition, I would rather have a broadleaf weed in my grove than a grasses, because grasses are like, for example, look at the root system of this thing. Most grasses have a very strong, especially if it's a perennial grasses, a very strong root system, which it's a real good sucker. It can suck up a lot of moisture and resources from the soil. So again, from our cover cropping, we were able to considerably reduce the amount of grasses and sedges, promoting a little bit of broadleaves, which was okay when in terms of what do you call that that competition perspective. Now, most importantly, we are seeing a lot of results now, but when we think about future, what's happened, and I want to bring in the concept of soil seed bank. I want to show you some numbers here. These are really big numbers. So what are they? The number of seeds produced by a plant. For example, weeds per plant. For example, black nightshade, amaranth, or guinea grass can produce this much of seeds per plant. And we have a model here. So technically, if they produce X number of weeds, it's going to be added into the soil, and that's the soil seed bank. And the research suggests that out of this, about 20% of the weed seeds are going to grow up as a new weed, right? So If you have more weeds in your grow, that is creating a soil seed bank, and that's a ticking time bomb. At some point of time, it's going to grow up, whether in your row middle or in your under tree area, and 20% of that is going to grow up. One thing I'm really excited about cover crops here is it can deplete the soil seed bank. For example, you're seeing a result now, but when you think about 5 years later, it is actually in the process of depleting that soil seed bank. And it's very evident from our data also. For example, in this figure, on the y-axis, we have weed density, number of weeds per square meter, and cover crop treatments. And we look at the consecutive planting. Like Sarah said, we had several planting per year. First planting, second planting, third planting, that even with legumes or without legumes, that weed pressure is reducing. That means less the weeds, less seeds added to the soil, less weed emergence in the next season. So long-term effects, cover cropping can contribute to that type of effects. So apart from that, I want to just touch base with something that I have seen when I go for data collection that I have seen in the our cover crop area, the biodiversity. All these cover crops we tested produce most beautiful flowers in the world. For example, radish flowers, the sun hemp, and these flowers are filled with floral nectarines and sugary syrups and pollens and things like that can attract a lot of good insects or beneficial insects. For example, here, see, look at a ladybird beetle, which was plenty in our cover crop area, eating an aphid, which can be detrimental for your tree crop like citrus, right? So there are plenty of benefits there. Apart from that, when we have a non-cover or regular area, grower standard, row middle vegetation, we always mow it. So frequent mowing is always needed, especially if you have grasses, skinny grass. And this frequent mowing is not good. I mean, if you mow more, the grass is going to grow more, and that can increase your water use potential in the growth. So having cover crop, actually keep it cool. Reduce the number of mowing. You don't have to mow, maybe 1 time, 2 times, that's all. And it's not grasses, it's suppressing the grasses. Apart from that, I have to say cover cropping is not entirely a chemical-free operation. We have to use herbicides from time to time, maybe for termination or maybe for land preparation and things like that. But again, when we compare to grower standard, where we use chemical application in between mowing, also known as chemical mowing, we can reduce the number of chemical application, and more details will be presented in the next presentation by Dr. Tara Wade and his team and her team. So, but I want to stop it here about that benefits, but definitely this is something that we need to keep in consider. So to summarize, to put it together, we found there is effective weed suppression, grasses and sedges. More importantly, reduce the grass and sedge pressure. Establishment is very important, but because we found the density can really affect the weed control. So the germination, the density, the lush growth of cover crop is very essential for weed suppression. And we are seeing the benefits today, and most probably we're going to see it tomorrow also, because the seed bank, soil seed bank depletion, cover crops are contributing to longer term control. And most importantly, the most important lesson that we learned is be patient. It takes time, as Dr. Davies, some of the Davies parameters discussed. Anyway, that was some citrus. We have some graduate students that also works on cover crops in several other production systems like vegetable weed management. I have my student Diderot. He has a summer cover crop going in the fallow season, then he plant vegetables, and you can see the difference here. Again, allelopathy and things like that may be coming into play here. There is less weeds in the tomato row middles when we have a summer cover crop. So that's all I have. I have to really acknowledge our graduate student Muriel Brewer and my biological scientist Robert River and also some weed science team. They do all the work. I just present. So I have to also thank my collaborators and grower cooperators, CRDF for funding opportunity. That's all I have. If I have question, I have take it. Otherwise, I'll be around. Thank you.
No questions.
Any questions from the?
Oh, hey, did you put in 3 separate cover crops through the year?
It was a mix.
Yeah, but you put them out 3 times.
2 plantings. We
2
had a spring/summer planting and a winter planting. Oh, okay. So two plantings per year, yes.
Okay, thank you.
All right,
Chris.
thank you.
Moving to the last presentation. Mainly about economics.
I'm Shourish. I'm working with Dr. Taraved. And as a part of this project, this bigger project, we are doing a cost benefit analysis of using cover crops in citrus production. So our primary objective is to provide a short-term analysis of using cover crops in citrus groves. So we do a partial budget analysis of using cover crops in citrus and then compare the break-even prices from using cover crops, compare the break-even prices to the historical prices in terms of dollar per pound solid and dollar per box using the historical data from FDOC. So citrus is very important, of course, in Florida, and citrus industry had in 2019 more than $6 billion of economic impact, then supported over 33,000 jobs. And also oranges are the top agricultural product of Florida with over $800 million in value and 51% of the total US production. Now, there are several challenges. I won't go over them because it has already been covered by the team members earlier. Challenges from pests and natural disasters. The key objectives of the industry, of the citrus industry, are sustainable production and profitability. So if you look at the past data, we see that citrus production has consistently decreased since the emergence of HLB in 2004-05 in Florida. And in fact, in 2017-18, California had a higher production of citrus than Florida, after which the Florida production has caught up. This is primarily because of the Hurricane Irma, and production and acreage have almost halved since 2005-06 in Florida. Now, there is no cure for HLB, and therefore one size fits all strategy will not work. However, there are ways to manage HLB. So there are various ways of managing HLB from the scientific point of view, and cover crops is a significant one of them, significant way to cope with HLB because it provides, it improves soil quality significantly. Now, so the direct benefits they provide, possible mitigation of symptoms associated with HLB, and cover crops primarily provide improved growing conditions that strengthen trees. Now, I won't go over the cover crop benefits because they have been extensively covered by the professors earlier. Now, what could be some of the cover crop adoption barriers faced by the growers? Now, there could be uncertainty regarding initial costs or owning or renting a specialized equipment such as a no-till seeder or no-till drill. And also, there could be an information barrier on what is the exact mix of cover crops to be used, because they vary by the grove. The soil quality or the growing conditions would vary by grove. Then others like benefits, sustainability, waiting period, and all. They have all been covered extensively earlier. Now, our objective is to start with the baseline costs of cover crops, which were calculated by Dr. Ariel Singerman. He is a professor at CREC, and he created the IFAS budgets for a 10-year-old grove. Now, to that average costs, we add additional costs of cover crops obtained from growers and account for savings from using cover crops. Now, the savings are short-term savings, 1-year savings. We will be looking at longer-term savings, say 5-year or 10-year savings later on, because those will be determined, for example, by the impacts on yield or quality later on. And also we have separate production costs for Valencia and non-Valencia oranges. Now, then for the next step, what we do is we calculate break-even prices in terms of dollar per box and dollar per pound solids. Then prices are calculated for yield, and quality scenarios. And these scenarios of yield and quality are in turn calculated from NAS and FDOC data. Now I'll go over the trends in the next few slides, and it's done for both Valencia and non-Valencia oranges. Then what we do is we compare the calculated break-even prices to the historical prices. Now, if the break-even prices are lower than the historical prices, there will be profitability of using cover crops in citrus production. And we obtain these historical prices from the FDOC field reports for dollar per box, dollar per pound solids, and then we discuss the scenarios and put them into context. Now, cover crop cost. So for the production, we have considered this as the per acre cost, which is around very close to $2,000. It's an average of 2019-20 and 2018-19 data from Dr. Singerman's studies, and then to that we have added other cost sources which we obtained after discussions with 3 growers who have used cover crops in citrus production. Now around 50 to 80 pounds per acre of cover crops are used for optimum germination. Therefore, I have used 65 pounds per acre, which is the average. Then the mix of legume and non-legumes, which are based on the studies conducted by Dr. Sarah and others in the team. And the average cost of the mix I got from the Hancock Seed Company, which roughly translates to $1.225 per acre. And then the total cost of seeds is around $80 per acre per application. Average. Yeah. So to that I have added, say for example, fuel cost, then labor cost, which I obtained from discussions with growers. And also the no-till drill seeder, I obtained the rental cost online, which is around roughly $20 per acre per application. And other costs are assumed to be around $5 per acre per application. And there are 2 applications per year. And so the short-term savings, no, total cost of seeds is no, this should be per pound. This is per pound. This is my mistake. Yeah. So if you multiply this to 65, if you multiply this to 65, then it's $80 per acre. So that's a mistake actually. Yeah. Sorry about that. Yeah. So 65 into 1.225. So 80 pounds per acre. Yeah. Sorry about that for the typo. And the short-term savings are considered from reduced mowing, as was discussed extensively by Dr. Ramdas earlier. Not mowing the cover crops would be very beneficial for weed suppression, and that's the only short-term saving that I've considered. I haven't considered, say for example, reduced herbicide usage and all, because I suppose those benefits will accrue over time. Yeah. So to this number, I have added these numbers that I discussed earlier, and total cover crop cost comes to around $220 per acre, and the savings from cover crops is 75.47, approximately $75 per acre, which is from reduced mowing, and therefore the net cost is around 2,133. So this is like 6.7% of total production cost, and or roughly 7.2 or 7.3% increase from the baseline cost. This, as I earlier said, this is a 1-year analysis, and 3 to 4 year analysis will include savings from reduced herbicides, net savings from changes in yields and quality, which will accrue later on. And also exact costs will depend on grove specific practices. Now let's look at the trends in yield and quality indicators before calculating the break-even prices. Now, if you look at the historical prices, this is for Valencia and this is for non-Valencia oranges. In terms of yield, the variability in yield is much higher than the variability in pound solids per box. And for Valencia oranges, there has been a consistent decrease since 2011-12. However, for non-Valencia oranges, it went down. Its minimum was in 2017-18 during the Hurricane Irma season, and after that it has been increasing. Also, in terms of boxes per acre, the yield has increased, and the yields are comparable. The last two, I would say 2018-19 and 19-20 yields are comparable to those from 2013-14 to 2015-16 through 2016-17 yields. And also the quality in terms of pound solids per box are comparable to those from 2015 through 2017 for both Valencia and non-Valencia oranges. Now, these are important because this will give us what will be the most appropriate scenario to compare the break-even prices to. Okay. Now, what we have done is using these historical prices, the last 10 years of last 10 years of yield and quality data, we have created 5 kind of quartiles with lowest, medium or median, and highest, as well as 25% quartile just from those 10 data points, and 75% quartile. This is for both yield as well as quality. Now, these are for various yield and quality quartiles. We have calculated the delivered in prices in terms of dollar per pound solids and in terms of dollar per box. Then after calculating those, we compare these prices to the price as week of delivery FDOC data, which again has been increasing, of course, since 2010-11 through 2019-20 for both dollar per box as well as dollar per pound solids. Then we do this for both the Valencia oranges as well as the non-Valencia oranges. Okay. So now if we look at this table, we see that at median quartile and median yield, at median quartile, both yield and quality quartiles, we see that, for example, the break-even price is around $2.23 per $2.23 in terms of dollars per pound solids. Now that price is comparable to the prices in the last 3 years of data, the last 3 years of price data, which is from 2017 through 2020. This indicates that at median, or like for example medium quality and yield scenario, it would be profitable to include cover crops in citrus production. Moreover, if you look at even minimum or quartile 1, which is we know that in 2019-20 season, the quality was so dollar pound solids per box was 5.9. So even if we are producing at 5.9 pound solids per box, at median yield, the price is 2.39, which is in fact lesser than the highest price recorded, which was in 2018-19 season. Now, so it could be profitable at the median yield scenario to include cover crops in citrus production for Valencia oranges. Now, if you look at the non-Valencia oranges, the effect is not as pronounced as Valencia oranges. Now, for non-Valencia oranges, it would be profitable to produce at the median yield and median quality quartiles, because the price is around 2.34, which is lower than the highest price that was recorded in the 2016-17 season. However, that price is higher than the last 3 years of prices that were recorded, like which is from 2017 through 2020. So therefore, including cover crops in production of non-Valencia oranges could be less profitable in terms of this analysis compared to including cover crops for production for Valencia oranges. So to summarize, net additional cost in citrus production with cover crops is around $144.53 per acre, which is 7.3% increase and around 6.8% of total production cost. And then at higher yield and quality quartiles, break-even prices are comparable to the pre-2016-17 season prices, which indicates that it could be profitable, or it would be economically feasible to include cover crops in citrus production. Now, this was in terms of short term, which is a 1-year analysis. In the longer term, say for example 3 to 5 years or more than 5 years, assuming cover crops improves quality by 1 to 2 quartiles from, say for example 2019-20 levels, or say for example between 2.7 to 7% for Valencia oranges, or 1 to 6% for non-Valencia oranges, corresponding break-even prices at median yield are comparable to the last 4 years of market prices, which are like including the increased yield and quartile and quality after like post 3 or 5 years of adoption of cover crops. Then profits at median yield quality scenario for both Valencia and non-Valencia oranges could be positive, since break-even prices are lesser than the peak prices that were observed in 2016-17 or 18-19 respectively. So to summarize, it would be economically feasible practice to include in citrus production. However, as Dr. Ramdas just mentioned earlier in his closing statement, like I guess producers have to be patient because the benefits accrue over time, and longer term benefits would be more pronounced than short term benefits, and longer term savings would be more would be higher, much higher than the short term savings. So so I would like to acknowledge CRDF as the source of funding, and then Dr. Sarah, Dr. Ramdas, Dr. Davie, Dr. Tarawed, and others for their guidance and help. So any questions? I'm happy to answer.
Any questions for Dr. Shourish? Wait, let's.
I'm a little confused. Davie said there was no significant difference in yield or yield quality.
Right.
So I'm not sure what you're saying about bringing prices and giving some kind of benefit to cover crops through the yield and fruit production.
Right. In the short term, there won't be any benefits. That's what I've done. I've shown the results for the first year of including cover crops. So I haven't assumed that there will be increase in yields or quality in the first year, but after 3 to 5 years of production, there could be increase in yields or quality, and it would be more sustainable to use cover crops in production.
But the research hasn't shown that yet.
No, Fred, I would call this like a what if analysis, like if this is the scenario, then you can make a profit, but growers will have to decide for themselves if this is something they can accomplish and if they'd want to use cover crops. But it's just the benchmark you'd have to meet for this to be profitable.
Right, but then Ramdas's presentation said that you could probably eliminate herbicide, not just
Right, yeah,
site
Yes,
costs.
yes.
yes,
Removed, but what I've done is I've kept the herbicide costs because herbicides are used in row middles as well as under the trees. So I've considered that they are used under the trees, but for row middles, because it's the first year, as Dr. Ramdas said, it reduces, it suppresses. So weed growth is like 15% of the original. I suppose maybe Dr. Ramdas can answer that better than me. So there will be some weed suppression, but in future there will be more.
Yeah, so I will add to what Shourish was saying. I think this is not going to change our under tree herbicide program, right? So this we are focusing on the row middle, and row middle, depending on the growers and the locations and the grows, there may be herbicides going in. Some are 100% are moving. Sometimes they bring in chemicals. Even if chemicals, it's a sublethal dose, very low rate of herbicides. So that's one thing. But again, here in cover cropping, as I mentioned, it's not a 100% herbicide free operation. It's a sustainable operations, but we have to bring in herbicides from time to time for plant preparation, termination, and things like that. So that's where I think the gray area is, whether we need to, whether herbicide cost is a big component is a question that we need to take a more look on. But anyway, this is not going to sit under tree program.
Any other question from the Zoom area? Thank you again, Shourish.
Thank you. There is some question, but this can be answered by Dr. Strauss or others.
If there is a question, go ahead and answer it.
So we have a question, how cover crops handle mechanical damage and equipment in groves and whether they can help reduce soil compaction. Very good question. We haven't done any actual quantitative measurements on this in this study. I will say, so when we plant the cover crops and the end of October, November, those continue growing through harvest. These were harvested in end of March, early April primarily. And yes, there was certainly some trampling of a lot of the cover crops with the goats going through. But as I mentioned earlier, some of that growth then rebounded a bit and managed to hang on, kind of depending on how hot it got, of course. But we would still see plenty of growth in the row middle where the tire tracks weren't going up until we would get really into the end of May when things really start getting warm and toasty. So I think they can handle farm equipment relatively well. And I know certainly operators have, driven their trucks during the growing season of these cover crops through periodically, and they don't seem to have any major issues with that. As long as you're not going up and down each row, 5 times a day, I think it's fine. Soil compaction is something very interesting. And I should mention that I have a new study that started that's focused more on soil health indicators and how we try and quantify changes to soil health. And it includes a pedologist from Soil and Water Sciences Department in Gainesville. And we are doing a lot of analysis, looking a little bit more closely at aggregates and bulk density and some of those soil physical properties and changes at different depths. And we're actually collecting soils down to about 50 centimeters deep. So really trying to get a better idea of how some of these things might be impacting not just the top foot or top couple of inches of the soil, but a lot deeper. And this goes back to the question of how do we really try and understand what are those contributions and measurements for soil organic matter and soil carbon and nutrient cycling. And so are these changes just on that shallow bit? Are they trickling down? And maybe that gives us a better idea of where some of these changes that we're seeing with some of the microbial activity might be impacting nutrients and nutrient flow to the trees in the future or in after longer term usage. That was a really long answer to your question, Brandon. I hope that covered it. Any other questions for anybody, any of the other speakers here too? That's the question was about endophytes within the trees and the tree roots. Something that I have not measured. It is something to think about. We are, as part of that other soil health study, we are going to start trying to really, one of the things we want to look at on the microbial side a little bit more closely is this idea of these plant growth promoting organisms, but which could potentially include some endophytes and things like that, to really try and get a, again, better idea of why some of these cover crops might be more beneficial and how are they changing some of those potentially beneficial organisms, but we haven't looked at that yet.
As far as improving food quality, we know that less fertilization could get it's very popular. Number one, food yield and quality.
So the question was about, yeah, the question was about whether there is any information or knowledge about whether the legumes cover crops might help reduce fertilizer cost and application and having less nitrogen improve bricks. We do not have any data on that yet. Again, the additional study that we're working on now is to try and get some more information on those questions. And I think there are some additional measurements that we're trying to make within the soils and monitor some of those nutrients a little bit more closely. I have a graduate student that would really like to do, some of that is very tricky to measure, and getting that complete nitrogen budget and how that is going into the tree is not the easiest task. And we've had a couple of discussions about ways to try and hone in on that, because that is certainly an interesting question. But yes, how much is remaining in the soil and how that might be impacting directly going into the tree is something we're still trying to assess. So the question was about whether there are cover crops that could help with phosphorus and water quality, and we have definitely not looked at that. I don't know if Dr. Kadyampakeni might have some thoughts on that, but like David mentioned, I mentioned, I think all of us, one of the big things that we've learned from this study is some folks have mentioned that there's still a lot of optimization to be done on trying to figure out what are the right species and what are the right cover crops to plant. And certainly, you know, we found daikon radish was a great one for the winter. That was clearly a home run, but other species, other times of the year, I think there's still a lot of room for improvement and a lot of room for optimization and lots of things to consider about what you want to put into your mix and what are the benefits, and certainly something that might help with phosphorus and water quality is something to consider.
And Sarah, just to add to what you said, on the phosphorus portion, I think we didn't see much change. I think what we noticed was most of the residual phosphorus in the soil. So I don't think at this point I can claim to say cover crops reduce the phosphorus loading or manage to eliminate that leaching of phosphorus to the groundwater. I don't have that information at this point. But one thing I noticed, I think for nitrogen, we saw that benefit of having elevated nitrogen, nitrates, even ammonia, where we did have the leguminous cover crops. But for phosphorus, we had hoped to see that benefit, but apparently we didn't see that over the course of the study. So maybe we can't speculate yet. Hopefully, what I'm speculating is that as we build the organic matter and we build the carbon, that might increase the ability of the soil to retain those phosphates and all those other nutrients and minimize leaching, but that will come over time. We had, I think we had an example on one of the sites in Lewisburg. There's, I think, a grower there, Ed James, who had shown us that I think he was able to build over organic matter over about 8 years, 4% to 8% of organic matter, but that took close to 8 years to get there. Yeah, so once you elevate the organic matter to some level, then those benefits come in, but we have to quantify that. Thanks.
Sarah,
Yes, sir.
I thought the USDA had data on buckwheat accumulating phosphate availability in Florida. And they recommended, I think it was a winter to use buckwheat, but it only lasts like 60 days.
Yeah, it's a really short crop. Yeah, that's what we've seen as
But
well.
I'm wondering if you can plant it twice in the winter down here.
That's certainly a possibility, and something we've had discussions about and was certainly on the table, was thinking about trying to squeeze in some more plantings. We were particularly thinking about trying to squeeze in another one in the summer, but things are just growing so well. We didn't want to spend the time and money mowing something to plant another one. And I think, again, in the winter, the trick is the timing with the rain. And so, but yeah, if you could try and squeeze in another planting in the winter of buckwheat, that could have some more benefits. You know, again, like I said, this is sort of our, you know, it's a 3-year study, but I look at it as a 3-year preliminary study, and that this is just trying to get started and see what are those first 3-year changes, what are we able to notice. But there's clearly a lot more work to be done, and lots of ways to try and optimize and customize.
There are actually not only the winter, but in the rainy season, because one species and the rainy season was another. These many organisms.
The question was about seeing differences in growth of the species. So we were planting different things in different seasons. So the daikon radish, we only planted in the winter. It doesn't like it in the super hot summers here. And even in the winter, if you planted it a little too early and we got another warm spell, it was not happy. The sun hemp certainly does not get as tall in the winter here as it does in the summer. In the summer, it, you know, can be as tall as me, and in the winter, it, you know, roughly knee height. But it still grows and it still produces flowers. So yeah, there are some differences in patterns, but we were also trying to tailor things a little bit based on what might do better. This is, again, an area where we're trying to do some more research now, and that the list of things that grow well here in the summer is much shorter than the list of things that grow well here in the winter, not too surprisingly. And so trying to find a balance of legumes and non-legumes has been a little bit challenging, but that's something that we're trying to work with some colleagues now on exploring different things. There was a question here about any studies planned for cover crops and cups. Not to my knowledge, but that would be cool. Any other questions? I know Dr. Kanissery is probably around for lunch as well, and Dr. Shourish and myself. And you're also more than welcome to contact us directly if there are any other questions.
Okay, thank you all for participating and for attending. Let's thank again the participation and also the presenters from here and from Lake Alfred. And if you are on Zoom and you are interested about CEUs, please contact Jean McAvoy or myself, send your name and email address and license number, and we will help you with the CEUs. Again, we need to thank Geo with Tropicana for sponsoring the program, and thank you all for participation. Okay, go ahead. Yeah, for those who are in Immokalee, we have lunch for them. For the others, hopefully we'll see you next month.